So let's talk about synaptic transmission. So we've covered a number of these syllabus objectives so far. So we're only up to this one now. Explain the passage of a nerve impulse in terms of synaptic transmission, referring to neurotransmitters, receptors, the synaptic cleft, vesicles, postsynaptic and presynaptic neurons, and signal transduction. So we've talked about the neuron and how the signal travels along the neuron as an electrical signal, as the action potential. And we talked about the direction of nerve conduction from the dendrites to the axon terminals. Now, at the end of the axon terminals, the electrical impulse cannot bridge the gap between or at the end of the axon terminal, so at the axon terminal, the neuron either makes a connection with another neuron, so the dendrites of another neuron, or an effector like a muscle or a gland. Now, whilst it's an electrical signal through the neuron, across this gap here called the synapse, it's actually a chemical signal. So that's what we're talking about now, is the signal across the synapse. So the synaptic transmission, this is the way in which neurons communicate with each other, but also neurons communicate with the effector as well. So the gap between the neuron, the gap between the axon terminal and the postsynaptic side is called the synapse. This whole area is called the synapse. And so it's quite simple that before the synapse, we call that the presynaptic neuron, and after, we call it the postsynaptic neuron. Whilst this whole thing is called the synapse, the actual gap between is called the synaptic cleft. And the signal that goes across is actually a chemical signal, not an electrical signal. So the action potential comes down and triggers the release of neurotransmitter chemicals in these things called vesicles. The vesicles then travel down to the end on the axon terminal, and through exocytosis they release the neurotransmitter, which then binds to receptors on the postsynaptic element, and that then passes the signal on. So here's a simplified diagram of all of these aspects. So this is the axon terminal of the presynaptic neuron. We have vesicles that contain our neurotransmitter. We have our gap between the pre and postsynaptic elements called the synaptic cleft. We have receptors that detect the neurotransmitter. So these vesicles get activated by the action potential. The vesicles move down towards the end of the axon terminal. Through a process of exocytosis, the vesicles release their neurotransmitter across the synaptic cleft to the receptors on the postsynaptic element. Just a little bit of a summary. The action potential cannot pass across this gap, but the action potential triggers the production of neurotransmitter, which travels across the synaptic cleft. Once it's received by the receptors, it either triggers a new action potential or produces a response in the effector cell. Interestingly, several poisons, including botulinum toxin or Botox, actually affect the function of neurotransmitters. So when the neurotransmitter is detected by the receptors on that postsynaptic element, whether it's a muscle or an other effector, or whether it's another neuron, it triggers a signal transduction. Basically, transduction just means changing the signal into another form. So it's basically taking a signal, interpreting it, and getting an action out of it. So the signal gets received and it triggers a response by the cell, but of course it's never easy. There's a series of steps, a transduction pathway that is required before the cellular response. But essentially, we just need to understand signal transduction is about receiving a signal and ultimately a response. Now, if it's another neuron, that response is going to be an action potential. If it's an effector, it's going to be related to hormones or related to a muscle contraction.